The gene that I decided to research is the TP53 gene. The goal of this research and presentation is to expand my knowledge about a gene that I'm particularly interested in. While I personally want to learn more about the TP53 gene, I also want to inform others about the differing functions and components that it involves. Cancer is one of the leading causes of death, and I think this gene is really important for everyone to be aware of. TP53 is a tumor suppressor gene that encodes the p53 protein that inhibits the growth and development of cancers and tumors. p53 functions to repair DNA in order to prevent altered or mutated DNA to be passed down to the daughter cells. When there is too much damage to be repaired, TP53 signals cells to undergo cell death, also known as apoptosis. The TP53 gene is mutated in a variety of different cancers. It's also known to be the most common gene mutation in cancer cells. All humans have 2 copies of the TP53 gene, which we randomly inherit from our parents. The TP53 gene is located on the long arm of chromosome 17, which is an autosome. It contains 11 exons spanning approximately 20,000 base pairs long, and it expresses Mendelian inheritance, which means that it's transmitted through either cell division, meiosis, or fertilization. The TP53 gene provides instructions for making a protein called tumor protein p53. The p53 protein acts as a tumor suppressor. It's essential for regulating DNA repair and cell division. It regulates cell division by keeping cells from growing and dividing too fast or in an uncontrolled way. The p53 protein is located in the nucleus of cells throughout the body, where it will bind directly to DNA. When the DNA in a cell becomes damaged, this protein plays a critical role in determining whether the DNA will be repaired or if the damaged cell will undergo apoptosis. p53 plays a role as a facilitator of DNA repair by halting the cell cycle to allow time for the repair machineries. It also directly impacts the activity of various DNA repair systems. The p53 gene provides protection from cancer development by maintaining genome stability. If the DNA can be repaired, the p53 protein activates other genes to fix the damage. If the DNA cannot be repaired, this protein prevents the cell from dividing and it signals it to undergo apoptosis. There are 2 types of mutations. There's heritable and there's acquired. Heritable mutations, also known as germline mutations in the TP53 gene, greatly increase the risk of developing breast cancer, as well as a variety of other forms of cancer. Li-Fraumeni syndrome is an example of this, and this is an inherited condition that is characterized by an increased risk for certain types of cancers. Acquired or somatic mutations are much more common than inherited mutations, occurring in 20 to 40% of all breast cancer cases. The cancers associated with somatic mutations are obtained throughout a person's lifetime and are only present in cells that become cancerous. These mutations change single amino acids in the p53 protein, which will reduce or eliminate the tumor suppressor function of the protein. And this is also an example of a missense mutation. This mutated protein cannot regulate cell proliferation effectively, so it is unable to trigger apoptosis in cells. As a result, damaged DNA can build up in the cells and they will continue to divide, leading to a tumor growth. The image below shows that there's roughly 380 codons, which also means that there's roughly 380 amino acids. This image shows that most mutations are affected in 6 amino acids. And those 6 amino acids are numbers 175, 245, 248, 243, 273, and 282. Thank you for watching.